Nice. Without further ado.
Thanks to everybody who comes, and I'd like to thank the organizer for this opportunity to share with you some of our recent results studying inflammation in human patients and animal models. Before I came here, I thought that I would introduce some of our motivations of why study inflammation, but obviously our first speaker has discussed this already. But we know that inflammation is the most common mechanisms in human diseases. For example, if we look at the leading causes of deaths in the US from heart disease, which our first speaker has talked about it, cancer, lung diseases, stroke, accidents, Alzheimer's disease, diabetes, liver disease, influenza, infection, and even suicide. In all these diseases, we know that inflammation is one of the most important mechanisms, if not the most important mechanism for that disease. So the system we chose to study is inflammation in injury, and the motivations are twofold. One is that for most of those patients that got into the intensive care units around the world, mostly before their injuries, they were healthy, so the baseline is relatively well defined. The second motivation, however, is a depressing one, which is that over the past half centuries of study of inflammation in injury, up to date, there have been more than 150 major clinical trials in this field targeting to block inflammatory response in critical ill patients, and unfortunately, every one of them failed so far. So back about 10 years ago, we started a consortium with more than 100 participating investigators from 17 academic institutions in the United States to study this disease. And we looked at the genomics, proteomics, as well as cell-level information of those trauma patients in the intensive care units in the United States. And then we compared the results with the results from murine models. There are obviously a lot of different type of results that have been generated so far. I wouldn't have time to get into most of them. So if you're interested, please visit our website. So the first thing I'd like to point out is that comparing to animal model studies, obviously studying patients, studying real patients in clinical setting is very challenging. And that's in a sense well known. There's a huge heterogeneity between individual patients in terms of their genetics, in terms of their lifestyles. And there's a diversity of the disease. In this case, different people get into injuries differently, and their gene response could be dramatically different between individuals. And the third thing that perhaps is less well understood is that there's huge variations in patient care. So, for example, we have showed that even by standardizing how patients are treated among different intensive care units, we can show that the death rate of those patients dropped by half. So the first study we did was to look at the gene response in white blood cells, because it's well known that the white blood cells are in charge of the immune response. And as we can see here on your left side is a typical heat map that's from controls to patients 12 hours after injury, up to 28 days after injury in the intensive care units. And each row is a gene. The red are those genes that go up, and the blue ones are those genes that go down. As we can see that there are clear waves forms after injury that can exist in our immune response. And if we just take a crude analysis of this group of genes. Let's say we take the upregulated gene. Let's say we take the upregulated genes on top and look at their gene functions. You can see all those typical pathways that have to do with innate immune response. For example, IL-6 signaling, IL-10 signaling, etc. And all the down-regulated genes have to do with adaptive immune response, as you can see on the right lower corner. All those genes have to do with T cells and B cell response. So immediately we can conclude that after humans get injured, there's a simultaneous upregulation of innate immune response and downregulation of adaptive immune response. Further, if we look at complicated patients, those patients that did not recover well, they stay in the hospital for more than a month, versus those patients that get out of the hospital in the first few days. What we can see at the gene level is essentially a more elevated inflammatory response and a delayed recovery. So we can characterize the first half where the gene first gets responded as the response time of their gene response, and the recovery time would be the time that the genes come back to normal. And we can see that essentially it's the recovery time that differ between the complicated patients versus uncomplicated patients. Obviously, we have to develop some of those tools to analyze this time course genomic data. I wouldn't get time to get into that. So the next thing we look at was the genomic response between different types of injuries. So here we look at patients who got hit by cars versus patients who got burned. So as you can imagine, those are two quite different types of injuries. So we look at the hundreds of patients, and the short answer is that it's a surprise to us that the gene response, as you can see in that scatter plot on the lower left corner, between burns and trauma in patients are quite similar. In fact, the R-square is more than 0.9, and 97% of the genes actually move to the same direction. So this suggests that there's perhaps a common inflammatory response to a variety of different acute inflammatory stimulations. So I just show you one example. Here is one gene, which is MMP8. And the x-axis is time after injury, and the y-axis is log2 gene expression value. And you can see that each dot there is a sample from a patient. And you can see that in both the trauma injury, which are the blue dots, the burn injury, which are the green dots, and there's this bacteria LPS study where we inject healthy volunteers with a dose of bacteria LPS to simulate infection. In both these three conditions, you can see an immediate upregulation of this particular gene within the first 2 hours after that stimulation, and the difference is basically the recovery time that's different. So the next thing we look at was how well do animal models of these diseases reflect human diseases. So to do so, we actually conducted experiments by choosing the most popular animal models for each one of these diseases, mouse burn, mouse trauma, and mouse endotoxin, and did the similar genomic studies on those animal models. And what you can see here is that comparing to the highly similar response in human patients between different types of injuries, the similarity in different mouse models is much poorer. And in addition, when we compare the gene response between humans and the animal models, the response is very low. And when we compare the human LPS model, which is model for infection, versus human patient response, you can see a reasonable correlation between the model and the patient response. Well, if you look at the mouse endotoxin model, there's basically no correlation with the human response. We can further look at 20 minutes? Okay. We can further look at the time course between human response versus mouse response. So these are human response, again, burn, trauma, and endotoxin, and you can see the similarities between the different type of human conditions versus mouse response of the same genes. And you can see that indeed, if you look at time course, the similarity is also very low. And without going into any details, it's just one gene that shows that the solid curves are human conditions, the dotted curves are mouse conditions, and they don't really correlate. And if, as computational people, if we actually characterize the response time, which are on the left, versus the recovery time, one of the additional difference that you would immediately see is that, so the line here is 1 day, is that for each one of the conditions, the response time is very short. It's within 1 day, first few hours, while the recovery time in patients obviously is typically much longer than the animal models. And if we actually look at the individual pathways, as we can see here, the solid bars are human conditions, and the hashed bars are animal conditions. And you can see that the correlation is actually quite dependent on the particular pathway that you're going to study. For example, I highlighted IL-10 signaling, where you can argue that there are some correlation between the animal models versus human conditions, while let's say if we take tau-like receptor signaling, the animal models, which are those hashed line bars, really reproduce human conditions very poorly. So we went one step further. We thought, whether we can actually sort of generalize our conclusions in injury to some of the other acute inflammatory diseases, for example, ARDS, which is acute respiratory dysfunction syndrome, basically acute lung failure, sepsis, and infections. So we look at some of the publicly available data sets that other groups have generated, and we compared the x-axis is the correlation of fold changes of genes, and the y-axis is the percentage of genes changed in the same direction. And you can see that on the upper right corner, the human conditions really cluster to each other, correlate with each other much better than the lower left corner, which are the animal models in each one of those conditions. So these are the results that I'm going to show you. So in the next few slides is just basically philosophical arguments. You can either agree with me or not. And I think there's increasing debates in the community, in the research community, on how to perhaps rethink the animal models. So the question really is, are mice really a miniaturized human? Because mouse models, as we all know, have been used as a dominant animal model in clinical, in biomedical research for the past few decades. So we thought about, why existing murine models poorly reflect human inflammatory diseases. I came up with 3 reasons. There are, probably other ideas out there. So one is essentially genetic distance between human and mouse. We know that roughly 75 million years have passed between human and mouse from a common ancestor. So we could argue that, mouse are just not human, and there is significant genetic reasons for that, that there's nothing you can do other than perhaps humanize mouse. And the second is ways that the existing animal models were developed, because most of the common models that were used were developed decades ago, where the molecular information, especially genomic information of mouse and human, were not that available. Therefore, a animal model was developed basically based on either the cause of the disease. For example, if we will study burned people, you just dip the poor mouse into boiling water, and you assume that the genomic response would be the same. But our argument basically shows that you actually have to do the experiment to see whether at the genomic level the response is the same or not. And lastly, perhaps the evolution of immune system is unique. And we actually know that our immune system is under constant pressure from the environment and ourselves. That's how our species get survived. Because up to perhaps 100 years ago, that's the only way that our species or our individuals can survive from either infections or other type of stress from the environment. And in fact, there are literature publications that show that perhaps for human, we took a strategy of resistance. We basically mount a battle against different bacterial infections and against different so-called danger-associated molecules, the DAMs, which are related to, for example, autoimmune diseases. While in mice, perhaps they chose another type of strategy to survive. They might choose to tolerate the bacterial species inside their body, for example, like a parasite. And therefore, they do not really mount a significant inflammatory response. One interesting area to study here is that actually, with modern medical care, would we actually want a significant inflammatory response, a so-called SIRS, systematic inflammatory response in our body, whether that's already not only not helpful, but actually has become very harmful to ourselves, and we perhaps should consider some kind of mouse-like response that's better for our survival these days. So one of the recommendations that we put out there is that since every drug compound to date acts on gene products, disease studies in animal models need to show that the molecular behavior of those key genes and pathways in those human diseases is actually reproduced in the models used. In other words, if the model actually does not reflect human disease, why would you actually study it, considering how many times in our basic research we have developed super animal models. We can perhaps generalize this into different other model organisms as well, although really important biological findings have been made in each one of those models, from perhaps bacteria to plants. However, when we identify candidates and try to make a link between those candidates to real patient care, we actually have to ask ourselves whether those findings can indeed reflect what's going on in human. So with the increasing availability of different new technologies, for example, high-throughput sequencing and other type of technologies available today, we think that perhaps instead of or in addition to conducting clinical trials based on existing animal model studies, maybe we should actually study human systems well and try to generate new hypotheses to improve our animal model systems. And one of the sort of additional approach that we could potentially use is that they are so-called ex vivo reconstitution of multiple cell types that are considered important in a particular disease process. Maybe that's another area that we could look into. And as computational people, we all know that we need a new computational methods to study human diseases in patients. And one of the areas that I think we all agree is, human studies are so complex. We're largely, measuring gene expression and protein data, metabolite data at the bottom of the ladder, while what we try to understand is a organ system level defect in disease process. Therefore, we actually need to somehow, computationally combine all this information across different layers and make sense of it. And obviously, there are a lot of experts in this audience that I hope we can talk more and follow up in this conference. So with that, I'd like to thank my group at Stanford and Mass General Hospital that actually did the work, and all the groups that are involved in this consortium. With that, I'd like to thank you for your attention.
How would you explain the weak correlation between the mouse burn and trauma studies? And did you use inbred or outbred lines for the...
Yeah, so these are two very good questions. For the first one, our thought was that basically these models were developed sort of independently. So, the mouse response to burns and mouse response to trauma could be indeed quite different. And one way to improve this is to essentially try out different burn models and try out different trauma models and see whether you can come out with one that's more like a human response. To your second question, that's also a very good question. And we know that among different species of mouse models, they are all quite tolerable to inflammatory response. For example, in terms of LPS response, there's a million-fold difference between human, the level of tolerance in human and mice. And, that's just really significant response. However, between different models, we see somewhere around 10-fold difference. So it seems like the species difference is much larger than the individual difference. Yeah. There are two responses. One is thank you for bringing this up. You're absolutely right that the mouse models, are very useful for variety of human studies. And your second point is also well taken, that humanized mouse potentially is one of the ways that we should try to improve the animal disease models. However, the part I like to disagree with you is that there have been tens of thousands, if not hundreds of thousands of papers published in this particular field using existing animal models. And that's a huge, in a sense, just to be blunt, waste of money and resource in the field. So while new animal models should be developed and should be explored, I completely agree with you, there is a huge problem of continue using existing animal models. And I hope that our paper would generate some of the discussions toward how would you actually best use animal models.
I have basically two questions. The first one is the coloration plots that you showed are based on the maximum fold change in the time course. Do you really expect that the maximum fold change between mice and human is the same, or is it not just more the direction? And the other question I have is where you had those public studies that you compared. I had a closer look at them. All the human studies were on the same Affymetrix array type, while the mouse experiments were on different array types. And there were also the sample types were different. Some of them were whole blood, some of them were just PBMCs, and some of them were white blood cells. Don't you think that the different array technologies in mice leads to a larger variation there? And don't you also think that the different sample types lead to a larger variation? So what do you think about that?
Yeah, that's a good question. So two responses. Well, first, regarding to the maximum change versus time course. So this is a time course that you can see, and you know, I didn't have time to go through that, but you can basically characterize different parameters of the time course, and no matter which parameter you use, as you can see here, the mouse response is different than human. The second response is that you're absolutely right that when you take, let's say, geo studies, there are going to be differences in the platform you used. Obviously, in this case, we compare the patients versus, not patients, the cases versus controls within the same platform. And the second is that we actually are in the process of conducting some of the experiments using exactly the same platform, and as you mentioned, that there's a cell type difference between each one of them, and these are unpublished results that we basically show that even you take the same cell type, for example here, monocytes, you still get a very low correlation.